All right, hello everyone. Welcome back to part two of our lecture for general biology. After last time, we talked about photosynthesis as a whole and the role of water and the nature of light and how that relates to photosynthesis. And now we're going to get into talking about the actual reactions, the series of metabolic pathways that lead from light to biological energy, storage of biological energy within sugars. So recall that this has two main stages: the light-dependent reactions of photosynthesis that involve the conversion of the light into biological energy, into ATP and H, which act as sort of carriers of energy, over to the light-independent reactions, the Calvin cycle, where that those carriers of the electrons, carriers of the energy, transfer that over to sugar, which is built up out of carbon dioxide, which is where all the mass of those sugars comes from. You can take a little moment here to think to yourself, like, how does photosynthesis happen when you don't have some of those conditions? If it's cloudy and there's less going on, think about how you might test this in a lab setting. We're going through this in our lab this semester. We can sort of experiment with different ways of simulating a cloudy day and how to put up some paper. We could use a sort of a sheet or something. You can simulate a further away sun, sort of modifying that solar intensity. But also the nature of the light. The light that passes through clouds is not the same in terms of its color either. So, photosynthesis is occurring within chloroplasts. What is kind of wild about the origin of chloroplasts, though, is these organelles were once cyanobacteria. If you look at the structure of cyanobacteria and what's going on within them, you'll see something that is very similar to the structure of the organelle that we call a chloroplast now. So there's actually been a lot of good evolutionary research done in sort of building this theory that is where chloroplasts came from, is they once existed as cyanobacteria that were symbiotically either ingested into or in some other way found their way into the cells of a eukaryote, and that eukaryote then, as it divided and reproduced, and it passed those chloroplasts on to the next generation of cells, and much more down the line, what we have are organisms that have what was once a cyanobacterium, has lost some of its properties. It has become an organelle of a eukaryotic cell, of plant and algal cells. So there are two layers of membrane to it. There's like an inner membrane and an outer membrane, and it consists of, within the chloroplast itself, there are these stacks, these thylakoids, which have each thylakoid is one of the layers in these stacks, sort of like a stack of pancakes. And then there's an inside and an outside, so we have refer to the stroma of the chloroplast as that's the fluid within the chloroplast, what was once the cytoplasm of a cyanobacterium. We refer to that as the stroma. And then within each of those thylakoids, each of those pancakes, you have the lumen, which is that space within them. So this is the organ in which glucose is produced. This is where the photosynthetic pigments, the chlorophyll, exists. All of the reactions of photosynthesis are occurring in different parts of the chloroplasts. In the case of eukaryotes, in the case of cyanobacteria, think of this as the whole cyanobacterium. Some of those, what occurs in the stroma of a plant chloroplast is going to just occur in the cytosol, the cytoplasm of a cyanobacterium. but we're very plant-centered in our discussion today. So, chloroplasts. I'll try to move my face out of the way here. But let's talk about the light-dependent reactions. So the first stage of this is another electron transport chain. So thinking back to the cellular respiration, how that ends in an electron transport chain where electrons are moving from one part of a complex to another, and then as they move along this chain, some work is done, some energy is transferred from one place to another. This is going to be a very similar sort of parallel story. But in this case, it's moving through a chain of proteins that we call photosystems. And each of these photosystems contains a molecule, contains chlorophyll, which is a pigment that is sensitive to particular ranges of light. When light of a particular wavelength hits it, that excites electrons and breaks up water. So this whole process actually starts with what was called photosystem 2, kind of called that because it was discovered second. So a bit misleading. There's some more readings here if you would like to check those out. I encourage it. But within photosystem 2, in most plants, two forms of chlorophyll are most common. They both absorb different wavelengths of red and blue light. They're sensitive to when they get hit by red or blue light. An electron on that chlorophyll molecule, which incidentally is kind of similar to a molecule at the center of hemoglobin, the protein that carries oxygen in our cells. That's an aside. But when they get hit by those particular wavelengths of light, because of the structure of that molecule, because of the size of that molecule, they get excited. One of the electrons on it gets excited. That gets transferred off to the next stage in this process. That leaves a space for an electron on the photosystem. That electron ends up getting lost from water. It gets stripped off of water. This is where the water comes into play here. And then that water gets converted over into oxygen and a couple of hydrogens. So splits apart that water molecule, and the oxygens join up together and get emitted as O2. You notice this is sort of inverse of what happens at the end of the electron transport chain in the case of cellular respiration, whereas oxygen coming in and accepting an electron here, it's water coming in to give up an electron, to lose an electron. So water here is getting oxidized, and then the photosystem water here is getting oxidized. It's losing that electron to the photosystem because that one electron got excited and went off and left. And this is all happening, by the way, in the membrane of the thylakoid, in like the membrane of each of these stacks of pancakes. So where does that electron then go? Oh, before we go on to that, think about here. So is photosynthesis going to involve hydrolysis or dehydration synthesis? Water is being used to break something up, or is water being lost? Is that right? No, that's not right. Correct answer, it should be hydrolysis. Okay, so where's that electron going? So that electron is then being used in the next stage to pump hydrogens, kind of like what happened again in the electron transport chain of cellular respiration. As that electron moves through different parts of this plastoquinone and this cytochrome, that creates energy that pumps hydrogens into the lumen side, the inside of the thylakoid, from the stroma, the inside of the chloroplast, but outside of the thylakoid. So that electron is going to pump some hydrogens. Sound familiar? Then it's going to get passed off to a protein called plastocyanin that will then carry it over to another photosystem. And here, something similar is going to happen where light is going to hit the photosystem. It's going to hit the chlorophyll molecule within the photosystem, and that electron is going to get excited again. So it's going to get excited. It's going to get passed off to an NADP molecule, which will combine with a hydrogen ion to form NADPH. That's one of our precursor molecules. That's one of our products of the light reactions. But then what we still have here is a whole bunch of hydrogens that have gotten pumped to this lumen side of the stroma. And then with our friend ATP synthase again is in the membrane of the thylakoid. And there's a concentration of hydrogen ions, just like with ATP synthase, that is found in the mitochondrion that helps produce the bulk of ATP in cellular respiration. This gradient of hydrogen ions can be used to drive the production of ATP. That should say ADP right there, not ATD. Yeah, this should sound a little bit familiar at this point. Similar sorts of structures. So viewed overall together, this is what's happening. That electron gets excited, gets passed over, that pumps some hydrogens. Then it gets excited again, it ends up on NADPH, and then those hydrogens then power this ATP synthase molecule, this ATP synthase protein. It's an enzyme. That's why it has an ACE. Embedded in the thylakoid membrane, that produces a bunch more ATP. So we've got NADPH and ATP produced thanks to light and the input of water. So, like last time, we want to make sure you have some notes of this yourself. Like we talked about with cellular respiration, one thing that's helpful here is to draw out these steps yourselves. Label where the light is being used, label where the energy is being produced, where the energy is at. Get creative with the colors, and have fun with it. Make something that you find useful to yourself as a way of remembering these steps. And now let's move on to the other reactions, the light-independent reactions. Not to say light isn't involved, because in order for these to happen, you need light to power the light-dependent reactions. These are powered by the light-dependent reactions, but these reactions themselves are not involving light. There's no photosynthetic pigments here, no photosensitive pigments. As a whole, we refer to this as the Calvin cycle. We split it into a few different phases. The first is this fixation of carbon, which uses 3 CO2 for each turn of the cycle. You get 2 turns of the cycle for each, like the full photosynthesis equation. But for 3 CO2 going in, with the help of this ever important enzyme called Rubisco, that all gets fixed together to this RUBP molecule. Rubisco, I say, is hugely important. It is thought to be one of the most abundant enzymes. It has different forms, but it's one of the most abundant enzymes in the world, because photosynthesis is the underlying metabolic pathway. Like, that's where most of the energy of life comes from, and all photosynthesis needs Rubisco to happen. Some version of the Calvin cycle is needed in order for photosynthesis to happen. So Rubisco, big important enzyme to remember. But anyway, with the help of Rubisco, that CO2 is fixed. Some ATP is used up to reduce it, in the process of carbon fixation, getting to this 1,3-bisphosphoglycerate. We enter the second phase, which is reduction, the gain of electrons from NADPH. And what you end up with is this molecule called G3P, which is the main output from this. This is sort of the precursor to sugar. This is essentially, there's some other parts to it, but this is essentially a half of a sugar, half of a 6-carbon sugar. It's a 3-carbon sugar with a phosphate. So that is one of the outputs, but then some of them end up getting recycled back with the help of additional ATP input. You regenerate this RUBP molecule. So then Rubisco can continue to act on new carbon dioxide, and the cycle continues. So that's why it's called the cycle. Just to review this, we're going to watch this video right here that sort of outlines this process again. Might be a little bit of reiteration, but just to sort of hear this from a different source. Let's pull this up.
Photosystem 1 and Photosystem 2. The photosystems transform light energy to chemical energy by exciting and then shuttling electrons from molecule to molecule in a chain-like fashion on the thylakoid membrane. This process is called an electron transport chain. Let's take a closer look to see how this process works. First, photons of light hit chlorophyll, a light-absorbing pigment in photosystem 2. Electrons in the chlorophyll are excited to a higher energy level. The excited electrons are passed to an electron carrier. Meanwhile, water splits and releases electrons. These electrons replace those lost at photosystem 2. The byproduct of this reaction is oxygen, which is eventually released into the air. The other products are protons, or hydrogen ions, which are released into the inside of the thylakoid, or lumen. The excited electrons move to the cytochrome complex. Some of the energy from the electrons is used by the cytochrome complex to transport additional protons into the lumen. The second electron carrier, a protein inside the lumen, receives the electrons and passes them to photosystem 1. These electrons have now lost most of the energy they gained from light in photosystem 2. Photons of light hit chlorophyll in photosystem 1 and excite the electrons again. The electrons are then passed to the third electron carrier. Finally, these electrons are either recycled or they interact with an enzyme and NADP+, the final electron acceptor of the light reactions, to form NADPH. Some of the energy from light is now stored in the reduced molecule NADPH. Some of the energy released from the transfer of electrons established a proton gradient across the thylakoid membrane. Protons that accumulated in the lumen diffuse into the stroma through an enzyme called ATP synthase. ATP synthase uses the potential energy of the proton gradient to combine ADP with inorganic phosphate to form ATP. In this way, the potential energy is transformed into chemical energy stored as ATP. ATP and NADPH now have stored energy from the light reactions. This energy can be used in the Calvin cycle. This light-driven electron transport chain is usually continuous in the presence of sunlight. It encompasses a series of chemical reactions that involve light absorption, energy conversion, and electron transfer carried out by the photosystems and other enzymes on the membrane of the thylakoids. The Calvin cycle takes place in the chloroplast stroma, the watery clear fluid surrounding the thylakoids. It's helpful to think of the Calvin cycle in 3 phases: fixation, reduction, and regeneration. In phase 1, inorganic carbon, in the form of carbon dioxide from the air, is incorporated into organic molecules, a process known as carbon fixation. 3 molecules of carbon dioxide react with 3 molecules of ribulose bisphosphate, RUBP, to produce 6 molecules of a 3-carbon molecule called 3-PGA. The enzyme Rubisco catalyzes this reaction. In the second phase, the organic molecules accept electrons, a process known as reduction. The 6 molecules of 3-PGA use 6 molecules of ATP and 6 molecules of NADPH, which store energy from the light reactions, to generate 6 molecules of G3P. The G3P molecules contain more electrons and are higher in potential energy than 3-PGA. 1 molecule of G3P exits the cycle. It can be used to make other organic molecules. In phase 3, the regeneration phase, a large set of reactions use the other 5 molecules of G3P and energy from 3 molecules of ATP to produce 3 molecules of RUBP. With RUBP reformed, the process can start again. Notice that in the Calvin cycle, the energy from ATP and NADPH produced in the light reactions is used to generate 1 G3P molecule from 3 carbon dioxide molecules. In this process, the electrons lost from NADPH are accepted by the carbons from carbon dioxide molecules, which are the ultimate electron acceptors of photosynthesis. G3P, the net product from the Calvin cycle, can be used to generate other organic molecules, such as sucrose or starch. Sucrose, produced by leaf cells, is transported through the vascular bundles to other parts of the plant, like stems and roots. Leaf cells can also sometimes form starch for long-term energy storage. Overall, the molecules generated by photosynthesis provide fuel and building materials that allow a plant to grow. Globally, photosynthesis produces an estimated 150 billion metric tons of carbohydrate per year and is responsible for the oxygen in our atmosphere,
all that carbon
making
dioxide
it one of the most
that's
important chemical
accepting the
processes
energy,
for life on Earth.
storing it in the chemical bonds of G3P and ultimately glucose and other organic molecules. That's where the mass is going. That's where the energy is going. Like that electron may have gotten excited in the photosystem after it had been donated by water. Eventually, that ends up on the NADPH, ends up on the CO2, ends up within the bonds of G3P. That is, if we're tracking where the energy went, that's sort of its path, and that's also where the path of the mass went. A lot of the water molecules aren't getting incorporated into the mass of the plant that is produced through photosynthesis. It's really coming from that CO2 that is being absorbed and captured in G3P and the other molecules from which that it's used to derive. So in incorporating, in order to do the Calvin cycle, Rubisco doesn't like to exist within certain environments. One thing is that in the presence of a lot of oxygen, you need to Rubisco will start doing other sorts of things. It'll start to oxidize. It'll be involved in the oxygenation of other molecules, and it won't be producing sugars in the same way that we need. In fact, it produces some molecules that are sort of toxic to the photosynthesizer, to the organism. So generally, plants need to have that constant exchange of oxygen and CO2. They need to be bringing in CO2 and they need to be releasing oxygen. The problem is when they're doing that, water is also always being lost. And like water, plants have a lot of different ways of dealing with that water loss, sort of maintaining an exchange of gases while also not losing too much of the water in the process. and I don't have time to get into the wild diversity of adaptations that plants have for this. They have like waxy coatings on their leaves. They have aerated roots to make sure that they're getting oxygen to the parts of them that aren't photosynthesizing, like their roots. They store water in different ways. They have different types of roots accessing that water to maintain that flow of water that we talked about previously when we were talking about transpiration. They need to be constantly maintaining that hypertonic environment that maintains a flow through the leaves or through the other parts of their body that's photosynthesizing. There needs to be a flow of water. But one of the ways that they do this is by opening and closing their stomata to let oxygen and CO2 exchange with the atmosphere. So CO2 flows in, water and oxygen flows out. And that transpiration is fundamental to the flow of water through a plant. But at a certain point, too much is not a great thing, because the plant still needs the water in order to donate the electron to the electron transport chain of photosystem containing those photosystems. So we need water. Plants need water in order to do photosynthesis. They also want to maintain a higher CO2 environment and not get oversaturated with oxygen, so where rubisco is not functioning well. So there's sort of physiological adaptations. Like I said, lots of them that I don't really have time to get into. But what I want to talk about here at the end of our lecture are the metabolic pathways that have evolved in multiple convergent cases. These have evolved in many different times, especially CAM photosynthesis. But there are three main types of photosynthesis. So far, we've really been talking about the simpler story of C3 photosynthesis, which is how most plants actually do photosynthesis, which is that CO2 is brought in. It's brought into the mesophyll cells, these cells that contain a lot of chloroplasts that are doing all the reactions of photosynthesis. They've got the chloroplasts, the Calvin cycle, and the light-dependent and light-dependent reactions are all going on within the chloroplasts of these mesophyll cells. So that is the way most plants do it. But there's a couple of different innovative ways that other plants have dealt with living in hotter and drier environments where you would otherwise lose too much water. If you need to keep your stomata open so that you get an exchange of oxygen and CO2, but you also don't want to lose too much water in the process. So C4 plants, these including we have C4 plants is a C4 photosynthesis is a form of photosynthesis in which plants have evolved means of sort of isolating certain processes so that CO2 can be maximized within the part of the cell where the CO2 is needed. So this includes plants like corn, by the way. Some of our other cereal crops, these like tropical grasses, is the sort of environment that these plants evolved in, these hot, somewhat humid, but like hot environments. In these conditions, what they do is they bring in CO2, and within these mesophyll cells, what first happens is these mesophyll cells are acting like a bit of an airlock where the CO2 is brought in and then it's immediately fixed so that it can be kept separate, and then the water loss is only what's lost from here. Then later on, when there are more favorable conditions for photosynthesis to proceed, when there won't be as much water loss, or when there would be more water loss, this 4-carbon sugar is then able to be brought into a different set of cells, these bundle sheath cells that are closer to the xylem where the water is. And using that, the Calvin that can be converted back into CO2, and then the Calvin cycle can be run. So it's sort of like a spatial isolation of some of these different processes of the light dependent and independent reactions. So sort of adding a buffer with a different type of cell and like sort of storing that CO2 in a form that is can be saved for later. Then there's CAM photosynthesis, which includes a lot of our cactuses, our agaves, our bromeliads, like our pineapples. These plants, a lot of these different plant taxa have evolved this form of photosynthesis where they completely do different parts of photosynthesis during the night versus during the day. So during the night, when it's cooler, they can open up their stomata. They can bring in CO2. They can let the oxygen out that they've produced through photosynthesis. They bring in that CO2. Again, they convert it into a 4-carbon molecule that can then be stored. And then they can close their stomata, convert that back to CO2, and then during the day, when the light is available to power the Calvin cycle, that reconverted CO2 thing then proceed through photosynthesis. So they do whole sets of reactions at night versus reactions during the day, rather than just always be going during the day when it's less efficient. So pretty wild how this is evolved in like multiple plant taxa, all dealing with like this problem of maximizing airflow while also minimizing water loss. So again, on your own, or if you have a study group, want to make sure you have a good sense of what the steps of the light independent reactions are as well, where CO2 is being used in the system. It's all going into the Calvin cycle, even if there was some sort of way of isolating that CO2 in time or space. And then ultimately remember to keep track of the energy, where it's being transferred, and where it's ultimately being stored in that G3P and in glucose molecules, other sugars as well. And last thing for today, there is a brief activity, a couple point activity for you to just review the processes of respiration and photosynthesis. This is structured as a small assignment that you should be able to see on Canvas, and I will talk about it a bit more in class, and I'll see you all in person.